Hello, everyone. Welcome to the first Marine Science Friday lecture of the season. Thank you guys so much for being here today. This lecture series is really focused on highlighting graduate student research at Florida Atlantic University's Harbor Branch Oceanographic Institute. And so throughout this series, I'm going to be introducing you guys to some of our students, and you're going to get a chance to hear about their projects and ask them all of your wonderful questions. Collaboration is really an important thing in research. It's integral to having a successful research project. So with that in mind, this series will pair two students together that are working on similar topics. Today's presentation is titled Talkin Turtles, Sea Turtle Health and Conservation, and our speakers are Marisa Pico and Skyler Klingshirn. They are both students in our Marine Science and Oceanography Master's program, working under Dr. Annie Page Cargin. And so I'm going to let them fill you in on all of the details about their research projects. We'll hear from Marisa first, and then Skyler will get a chance to tell us about her project as well. And of course, like I said, we want to hear your questions. So you can feel free to ask your questions by using the Q&A button that's the bottom center of your screen, and we'll answer all of those questions towards the end. And you can ask either student your questions.
Hello. My name is Marisa, as Madeline said, and I'm part of the Page Cargin Lab. And today I'll be talking about sea turtle health and conservation. So sea turtles, there are seven species of sea turtles, and they are located globally. To start with, we have the leatherback sea turtle on the left side of your screen, and they're a pelagic species, and they're found in the open ocean. We have the Kemp's Ridley, which is right next to the leatherback sea turtle. They're the most endangered type of sea turtle, and they're found in the Gulf of Mexico and along the northeast coast of the United States. Following the Kemp's, we have the loggerhead sea turtle. They're found on the southeast coast of the United States and in Central and South America. Right below the loggerhead, we have the green sea turtle. They're typically found in warmer waters and near shore, bays, lagoons, and reefs. This is basically because they eat seagrass, and bays, lagoons, and reefs are the main habitat for seagrasses. To the right, we have the olive ridley. They're usually found around African coastlines and the Pacific and Indian Oceans. Right below that, we have the flatback sea turtle, and they have only been seen around Australian coastlines. Finally, we have the hawksbill sea turtle, and they're primarily found in the Indo-Pacific. So because sea turtles are global species, there are global threats to them. On the left, sorry. So there are local threats to them. So around Florida waterways, they experience this thing called fibropapillomatosis, which is a herpes-like virus, and it promotes tumor growth both internally and externally. As you can see in the left-hand picture, those pink things on the face and around the shoulder are the FP tumor growths, and these can be fatal as well. On the top right, you see a boat strike wound. So around Florida waterways, there's a lot of boat traffic, and it does cause wounds, some which can be healed, but most of them are fatal. The bottom right picture showcases harmful algal blooms. So this doesn't directly harm the sea turtles, but indirectly it does to their food source. So usually harmful algal blooms accumulate at the surface of the water, and they do not let sunlight penetrate through the water. So the seagrass ends up dying off. And that can evidently hurt sea turtles and their main food. Because of these local threats, the Page Cargin Lab has been doing sea turtle health assessments. Sea turtle health assessments are an integral part of determining a population's overall health and recovery efforts. And in these health assessments, we go through a variety of procedures, starting with body morphometrics. As you can see in the top picture, we take measurements of the carapace, and we also take weight, as well as clinical blood data. So on the left center picture, you can see how we draw blood from the dorsal part of their neck. And then we sometimes do scoot and skin samples depending on if there's an isotope analysis research being done, which you can see on the center right image. And then finally, we do a body condition evaluation. So that encompasses visual subjective body condition score, calculated body condition index, both of which I will talk about in the further slides, and ultrasonography, which is part of my research. And you can see us doing that on the boat in the very bottom picture right there. So to begin with, body condition score is subjective. So there happens to be a lot of inner variability between evaluators. It's based on a ranking system, 1 to 5, 1 being emaciated, which would be the turtle in the bottom. You can see both the dorsal and ventral view. And then a robust turtle would be like the turtle in the top right. So some ways to determine what number they are in the ranking system is by looking at the fat abundance around their neck, their shoulder, and their hind flippers. And you can look at basically their eyes. So if their eyes are sunken in, or if their plastron is concaved, then that is a sign of emaciated turtle. So in the bottom left picture, you can see how the neck and shoulders nearly have no fat, and their eyes are all the way sunken in. And on the right, you can see how the plastron is concaved inwards, as opposed to being flat or extended outwards. Body condition index is basically like calculating your body mass in humans. And you do this by using the equation on the screen. And to do this equation, you need the straight carapace length, which is what they're doing in the top picture. And you need a mass, which is what they're doing in the bottom picture. And you need this mass in kilograms. So this is the more precise way of calculating their body condition. However, it can be a cumbersome task for weighing turtles that can, are about hundreds or thousands of pounds, such as like a leatherback sea turtle. Also, when you're in tight spaces on a boat, it could be really cumbersome as well. So that's where my research comes into play. We want to validate ultrasonography as a novel approach to estimating body condition in sea turtles. So this is ultrasonography is widely used in the medical field, both in humans and in animals. As you can see on the screen, it's been used on horses and dogs as well. And very few times has it been used on sea turtles. There's only actually one study that has shown to validate ultrasonography in sea turtles, and that is on the leatherback sea turtle. So the study in which they validated ultrasonography as a method to estimate body condition was on free-ranging leatherback sea turtles. And this is a perfect study for them because they can weigh up to 1,500 pounds. So measuring something as big as that could be really, really difficult. And what they did was they used ultrasound-assisted measurements of subcutaneous fat in live and dead leatherbacks. So in the top right picture, you can see them putting the ultrasound transducer on the dorsal shoulder region. And then the picture at the bottom basically shows what they see in the ultrasound. So the dark region labeled B is the subcutaneous fat that they're measuring. And then to validate that that measurement is correct, they paired it with direct measurements from surgical biopsy and necropsy. So the biopsy would come from the live turtles and the necropsy would be for the dead leatherback turtles. As well, they paired it with histological measurements as well. And as a result, the dorsal shoulder region proved to be the most consistent and easily obtainable images with ultrasound. And then that's where my research comes into play because this has only been done on leatherback sea turtles. So as a result, I will be doing this on green and loggerhead sea turtles. So my objective one is to use and compare two different portable ultrasound machines to measure subcutaneous fat depth in green and loggerhead sea turtles, both of the sea turtles that we find primarily around Florida's coast. Objective 2 is to determine the anatomic location or locations that deliver the most consistent images. And to do this, I need to collect sea turtle carcasses. We have collected 10 loggerhead sea turtle carcasses, 5 of which are juvenile or subadult, and 5 of which are adult, as well as 10 green sea turtle carcasses, 5 of which are juvenile or subadult, and 5 of which are adult. And then these carcasses come from turtles that have stranded. So to help facilitate the transfer of these carcasses, we've had help from Florida Fish and Wildlife, Loggerhead Marine Life Center, Crow Rehab Center, Brevard Zoo, and Ecological Associates. So the methods to this research starts off with obtaining body morphometrics to be able to calculate that BCI. So you need a straight carapace length and you need the mass. So you can see in the top picture how we are taking the straight carapace length, and then on the bottom left, it shows how we actually weigh these turtles with a crane. Moving on, we will be comparing 2 different ultrasound machines. So the Renko lean meter and the Ibex Evo. The Renko lean meter is typically used on livestock and it costs about $650. This machine just delivers one simple number and it's measured in millimeters. It also does not measure anything under 3 millimeters, so that's important to note. On the other hand, the Ibex Evo is veterinary certified and costs approximately $24,000. So there's a huge difference in price when it comes to these 2 ultrasounds. Another big difference is that the Ibex Evo provides a visual image of the subcutaneous fat and has internal calipers that help us to measure the fat of the sea turtles. So using the 2 ultrasounds, we will be measuring 4 anatomic regions on the sea turtles, starting with the lateral neck, which is seen in orange, the dorsal neck, which is in blue, the dorsal shoulder region, which is in green, and the dorsal base of the hind flipper, which is in purple. And at each anatomic region, we'll be positioning the turtles in 3 different ways, starting with neutral, which is how the turtle would naturally lay, extension, which is the flipper at a 90-degree angle, and flexion, which is the flipper parallel to the body. So starting with the Renko lean meter, it's very simple steps. You start off by putting coupling gel on the probe. Then you place a probe at the area where you would like to measure. You start off by holding the button, pressing the probe against the skin, and then you just read off the number, just as simple as that. So in this case, it was 5 millimeters deep. On the other hand, the Ibex Evo is there's a bit more to do, but very similar. So you put coupling gel onto the transducer, place the transducer onto the skin where you would like to read the fat depth, and then you look for the best image and you take a screenshot. Then you would use internal calipers to measure that subcutaneous fat. So in the image on the right, you can see how there's a dark black region in between 2 white regions, and that is the subcutaneous fat. The green line just shows the calipers that I used to measure. So that spot right there would be about 0.47 centimeters deep of fat. And then after using both ultrasonographies, we do have to compare it to a gross measurement. So on the left, you see that we took a biopsy of the area that I ultrasound, and then I use a ruler to manually measure the fat depth. Then after measuring it grossly, we put it onto a slide and stain it so that we can read it histologically underneath a microscope, which is the image on the right. So you can see how there's a lighter pink area, which is signaled by the red line in between the skin and muscle layer. And that's exactly what we're measuring to compare that and validate that the measurements we're getting on the ultrasound are correct. So for objective one, we're doing a data analysis. Currently, we've acquired all of our measurements and we're in the process of analyzing and comparing the measurements. We will be doing a method comparison between ultrasound machines, gross measurements, and histological measurements. The stats that we will be doing is the Bland-Allman plot and passing Bablock regression, which determine the limits of agreement. So basically, we want to compare the Renko lean meter number to the IBEX EVO measurement, to the gross measurement, which we took with the ruler, to the histological measurement. So it's important that we compare it to as much as possible to make sure that it's all accurate. After that, we will do simple linear regression to show associations between the calculated BCI and ultrasound-assisted fat depth measurements. Objective number 2, which was to see which anatomic region had the most consistent images. We will take ultrasound images at 4 anatomic regions that I mentioned before at the 3 different positions. And we will see which region delivered the most consistent images. And as predicted, the dorsal shoulder region would consist of most consistent images. And the image on the right is one of the images of the dorsal shoulder region that we took of an adult green sea turtle. And as you can see, it's very thick. That dark black region is about 1.21 centimeters. So it's really easy to tell from that, as opposed to the hind flipper, for example. So this same turtle, the adult green, this is the image that I got for their hind flipper. So it just goes to show how that turtle was really fat, but you can barely tell based on the hind flipper. So the dorsal shoulder region would present the most consistent images. So some future projections. The point of this study is so that veterinarians and sea turtle biologists can quickly estimate body condition under tight time constraints or space, for example, on the boat. And some other projections that I would like for the study is that to validate that ultrasonography work in other sea turtle species. So this can be more of a common used method to determine body condition. And also it can help create management and conservation efforts as well. So with that, I will turn it to Skyler, who will tell you about her research.
Thank you, Marissa. Hello, everyone. My name's Skyler, and today I'm going to be talking to you about my research with the leatherback sea turtle. So the leatherback sea turtle is the largest living turtle, with adults weighing over 800 pounds and nesting females that we see on Juneau and Jupiter's Beach, Florida, averaging around 5 feet in curved carapace length, which is just the length from the top of the shell to the bottom of the shell. So leatherback sea turtles are found globally, which you can see on this map. They have a very wide range, and throughout that range, there are 7 distinct populations. I'm going to be focusing on the Northwest Atlantic population, which is shown by the red circle, and this population is considered endangered. Specifically in Florida, from the bottom graph, we can see that the nests that we've been seeing in Florida has decreased. So from 2008 to 2007, the average amount of nests that we get each year on Florida beaches has been declining. So there has to be something going on that is preventing these leatherbacks from maintaining the normal nest counts that we used to see. And this is concerning because leatherbacks are very important. While they are very massive, they only feed on jellyfish. So because they are so big, they have to eat a lot of jellyfish. So leatherbacks help keep the food web balanced. If they weren't there to predate on the jellyfish populations would boom out of control, and that would result in many more jellyfish preying on their food source, such as zooplankton or fish larvae. So if there were to be too many jellyfish, the zooplankton and fish larvae populations would decrease, which would result in a cascade of negative effects for the food webs. So it's really important that we have these leatherbacks out there feeding on jellyfish and keeping the balance in the food webs. Also, the eggshells and unhatched eggs that are left on the beaches after a nest hatches can provide nutrients to that environment. And this is a more sad thought, but some of the hatchlings that don't make it are also a food source for other animals. So leatherbacks play an important role in the ocean in many ways. However, with many sea turtles, there's lots of conservation challenges with certain life history characteristics. So leatherbacks have late sexual maturity. Specifically, the Northwest Atlantic population reaches sexual maturity around 24.5 to 29 years of age. So if you have a population that's declining and you want to increase those numbers, having to wait nearly 30 years for an individual to be able to reproduce can present some challenges. Also, they are a highly migratory species. They're the most pelagic species, meaning that they spend a lot of their life in the open ocean. So it's really hard for us to learn about them when they're so far away. Their migration is really important because they can interact with a lot of threats at that time, and they do this migration all the time. So we know that adult and subadult leatherbacks will migrate from their foraging grounds off the coast of Canada for the Northwest population, and then they travel through the open ocean to the tropical waters for their nesting grounds and back. We know that mature females nest every 2 to 3 years. So every 2 to 3 years, they're making this trip from the northern foraging grounds to the more tropical waters. Males, on the other hand, will migrate to the nesting grounds to breed with the females, so they can make this trip every year. And then the subadults are thought to make this trip to increase their foraging opportunities. So like I mentioned, when they're making these migrations, they can encounter a lot of threats. One of those threats that we know that they encounter are vessels. So like you and I, sea turtles have lungs, so they must come to the surface to breathe. And what else is at the surface? Boats and propellers. So they can interact with these vessels. The threat of vessel strikes increases in shallow and coastal waters, like the waters that they have to travel through when they're nesting. And this is especially worrisome for reproductively valuable life stages, such as the adults and the subadults. They don't have a very high chance of survival, so the individuals that actually do make it to adulthood are very valuable. So we want to make sure that we're keeping them safe. We've also seen that vessel strike injuries increase proportionately to the registered boats. So the more stranded turtles that we see with vessel strike injuries increased as there were more boats being registered for Florida waters. So this is just a real life example that I've seen in my research. So the picture you see here is of the carapace of a turtle named Panda. We saw her on May 1st of 2019 to lay a nest, and you can see that there are some small lacerations on her carapace, but then just 20 days later, when she came up to lay another nest, we see these really deep parallel lacerations, which are typically indicative of a boat strike. So while she was in between having clutches ready to be laid, she was unfortunately struck with a boat. So we know that this is a problem that these animals face. Another huge threat that they face are fisheries. Specifically, longline fishing is an issue. So in the top right, you can see a schematic of a longline fishing line. So there'll be just a very long line attached from a boat that has a bunch of baited hooks, and these hooks will sit in the water for hours so that the fish that the fishermen are targeting will try to eat the bait, and then they can reel them in. However, other animals, such as turtles and even sharks, can interact with these hooks negatively. So sometimes a turtle might accidentally get the hook in its mouth or on its shoulder, or they can also get tangled up in this fishing line. So a study that looked at nesting and foraging leatherbacks in the Northwest Atlantic found that fishery-related injuries accounted for the highest proportion of anthropogenic injuries. So you can see on this graph that there were the most injuries due to ropes, lines, nets, or hooks, even compared to boat strikes. So we know that fisheries are a huge issue. And while disentanglement is encouraged, there's a very high re-entanglement rate. So even if you get to a leatherback and you're able to set it free, it's very likely that it'll get tangled up again. And a lot of the times you don't get to leatherback in time. Since I said that they have to surface to breathe, if they're trapped in these lines for too long, they could drown. Another study looked at the long-term susceptibility of leatherbacks interacting with longline fishing gear. So essentially what this study did is they tracked leatherbacks and found the areas where they occupy the most, and they overlapped that with areas that have very high fishing pressure. So when you put those together, you can see areas of low, medium, and high susceptibility of leatherbacks to interact with long lines. So the red areas are areas where there is high fishing pressure, and also they saw a high density of leatherbacks using those areas. So along the continental US, you can see there's both high and medium susceptibility for leatherbacks to interact with these fisheries, which is very bad since they come to these areas to nest. So we know that they interact with these threats through injuries that we see when they come up to nest. So on the left here, you can see there's a really deep laceration on the shoulder, which is likely due to a fishing line tangling the flipper. In the middle, there's 3 very deep parallel lacerations, which are most likely from a boat propeller, as well as there are social or natural causes, such as predation. On this turtle's head, you can see shark rake marks from the teeth of a shark. So we know that these threats exist because we see them exhibiting injuries when they come up to nest. However, they're not regulated. So we know they exist, but we don't have a lot of regulation to help out these animals, and we need a way to quantify these. So there is a need for continued research, especially on these human-turtle interactions, to help better protect these animals. Along with just visual cues to determine the health of the animal, we can use blood parameters to be more indicative of how the animal is doing. So things such as packed cell volume, white blood cell counts, and total solids can be measured because altered values compared to the normal baseline values that we would see can indicate this turtle is sick or has a blood disorder. A previous study has looked at the blood parameters of nesting loggerhead sea turtles and found that there were altered values compared to the injured and non-injured turtles. So there's never been a direct comparison of the health of leatherbacks that were injured or non-injured, which leads me to the research objectives of my project. So the first objective is to determine the prevalence and extent of anthropogenic and natural external injuries on leatherbacks nesting in northern Palm Beach County, Florida. And to 2, investigate the relationship between injuries and leatherbacks' health based on these specific blood parameters. So working with Loggerhead Marine Life Center, I conduct surveys along the Juneau and Jupiter's beach. So in 2019, Palm Beach County represented 36% of all the leatherback nesting activity in Florida. So this is a really good area to be a gauge for how this population is doing. So my survey zone starts at Jupiter Inlet and goes 9.5 miles down to John B. MacArthur State Park. This area was surveyed April 1st through July 1st in 2019 and 2020. We monitored this beach starting at 2100 and went till 3 a.m. or later if necessary using ATVs, and we were able to complete these surveys 4 nights a week during the 2019 season and then 7 nights a week during the 2020 season. So just to show an overview of the nesting process, because it's pretty cool. Hold on, this one's not muted. So when we're out on the beach, what we're looking for are these massive tracks. When a turtle is ready to come up and lay her nest, she'll crawl up onto the beach, making these tracks, which can span 6 feet in width. So it's really hard to miss them. And then she looks for an area that she likes, and she'll start using her hind flippers to dig an egg chamber, which is shaped kind of like an upside down light bulb. And then she'll lay her clutch of eggs. And when she's done laying all of her eggs, she'll use those hind flippers again to pack the sand over top of them to protect them. And then she will take her front flippers and throw sand all over the place to disguise the whole area so no one can find them. And then she makes her way back to the water and waits until she's ready to lay another clutch. And then in about 2 months, we will have hatchlings that are making their way to the ocean. So the whole nesting process is really important because like I said, they're pelagic animals, so they spend a lot of time in the open ocean. So when they actually come up on the beach, it's a really great opportunity for researchers to learn more about them. So for my surveys, I opportunistically encounter leatherbacks. And once she starts laying her eggs is when we approach her. We don't want to disrupt her as she's getting ready to lay her eggs. Once she starts laying, she goes into kind of a natural epidural where she's not very aware of external stimuli. So that's when it's okay for us to approach her. And so the first thing that we do is we will identify her either through flipper tags or through PIT tags, which are just an internal tag, which we can use the scanner to identify who she is if we've seen her before. Next, we will take a blood sample in these vacutainers, and then we measure the turtles. And the last thing that we do is look for any external injuries. So we do a whole analysis of their external body, looking at their head, their neck, the front and rear flippers, as well as the carapace. So if we do see an injury, we'll make note of what body part it's on. We'll also make note of the stage of the injury. So if it's fresh and bleeding, or if it's starting to heal, which we can tell by yellowing skin, or if it's completely healed and has a pink scar. We can also assign it a presumptive injury source based on certain characteristics that have been written about in previous literature, and we'll also measure it. Through FWC, we are permitted to take flash photographies of all injuries. So I'm using these photographs and working with a variety of other sea turtle experts to help better validate the presumptive injury source. It can be pretty subjective to determine what caused each injury. So by having more people review these injuries, we have a better idea of what actually happened. And then after we complete our surveys at 3 a.m., we go back to the lab and we processed all of our blood samples. So the first thing we do is we take a sample of the whole blood and smear it to make a slide, which is then dried and stained. And through microscopy, we can estimate the total amount of white blood cells. And then the rest of the blood is centrifuged to separate the plasma from the rest of the cellular fractions. So on the bottom, there is a small centrifuge tube, and we can use a hematocrit reader card to determine the packed cell volume, which is a percentage. And then we take a small drop of plasma and put it on a refractometer to determine the total solids. So just some summary data of what we've seen the past 2 summers. In 2019, we encountered 51 individual female turtles, with 42 of those individuals exhibiting at least one injury. So the total injury prevalence was 82.4% of all turtles, of all leatherback turtles that came to nest had at least one injury. We documented a total of 129 injuries, so that leads us to an average of 2.53 injuries per turtle. In 2020, we had a much busier year, which was really exciting. We encountered a total of 111 individuals, with 93 of them exhibiting at least one injury. So that leaves an injury prevalence of 83.7 of the turtles had at least one injury. We documented a total of 279 injuries, leaving us with an average of 2.51 injuries per turtle. I'm still in the process of analyzing all my data, but for objective 1, I'm going to be determining the prevalence of each injury source and type. So I'm going to be comparing if there's differences between the injury source categories, mainly trying to answer the question, is one source more prevalent than any others? So for example, if we saw significantly more fishing related injuries compared to boat injuries, we know that fisheries are the main threat that they face, and we really need to work on improving our fisheries management. And for objective 2, to determine relationships between the health variables, I'm going to be looking at the turtles in different groups. So the turtles are grouped by the type of injury that they have, the stage of their injury, and the source. And so mainly I'm trying to see if there's any altered blood parameters related to the certain injury types, stages, or sources. So if we can find out relationships between what might be reducing the sea turtle's health, if there's a certain source that has significantly lower or less healthy blood values, we know that we need to, again, focus on that source. If we know what's hurting them, we can start to fix those issues. So for some future work, we're going to keep quantifying these injuries and their potential impact to better inform management. We can also compare what we see on our beaches in Florida to other nesting beaches. Since they are globally distributed, that's one of the main challenges we face as conservationists, is trying to come to one consensus of how we should protect these animals. So by comparing from beach to beach, we can get a better idea of the threats that they're facing all over the world. And we can even tie in some of Marissa's research looking at body condition and injuries and see if there are certain injuries that result in lesser body conditions compared to others, and as well as always inform people. So you guys are all experts now on sea turtles, so you know you can be more mindful when you're boating and clean up your fishing gear and even make more informed seafood choices. So choose seafood that wasn't caught in a method that could potentially harm sea turtles. So with that, we will take any questions.
Okay, that was great. First of all, I'm gonna applaud both of you for the entire audience, because I know that when we are doing a virtual chat, you don't get that reward of a nice applause. So I got you. And then we can dive into some questions. But did you guys have anyone that you wanted to thank maybe that has helped you guys out with your research? I know that it's a little awkward having two separate people doing their talks, but I know usually that's the last slide of a presentation.
Well, first and foremost, our advisor, Dr. Annie Page-Karjan. She's led us both in great ways and influenced us a lot, and taught us a lot about sea turtles, more than we already know. And then just all of the people that I've collaborated with, all the organizations I mentioned in my presentation, and all of our lab mates who have helped us with our research as well.
Great, awesome. And we do also have their advisor, Dr. Annie Page-Karjan, here with us today. So she can help out and answer questions if needed, but I think that Skyler and Marisa should be able to handle most of them. So again, a quick reminder for you guys, if you do have questions, use that Q&A button bottom center of the screen, and we can answer those. So our first question is from Nolan. I don't know if either of you guys know him. He says, thank you, excellent job. What is the most stressful part of gathering data of a live sea turtle in the field? So that one's for Skyler, I'm guessing.
Yes, I can definitely answer that. Thanks, Nolan. So I think one of the most stressful parts for me is making sure that we get everything done while she's laying. Like I mentioned, when she's actively laying, she's not really aware of what's going on, so we know we're not really disrupting her. And sometimes they lay really fast, and you want to make sure you get all the information from each turtle. So you want to get the blood sample, the measurements, make sure you look for all the injuries. So each one is so valuable. So it's kind of, once they start laying, you got to get to work and get all this information. So I think that's the most stressful part, is just making sure you get everything done in the small amount of time that you have with each turtle.
I couldn't imagine, although I'm not a night person, so I would not be in the middle of
I love the nighttime. It's so much fun. And with the leatherbacks, they're so massive. If they start covering or moving to that next stage of nesting, we got to watch out for their front flippers. They're super powerful. You don't want to get hit by one.
Have you ever gotten like covered with sand while they're
I've definitely had sand thrown in my face. Absolutely. But that's okay. We still got the information.
That's good. So the next question is for Marisa. It is, do you know the cause of death of the turtles that you were doing your research on?
So most of the turtles just strand, but I would say most of the cases are a lot of boat strike injuries. There's, I think, one turtle that I can remember that the sand dune fell on it as it was laying its eggs. So it basically suffocated. We found some sand inside like the throat. And there's also a lot of turtles that we don't know from just looking at the outside how they died. So that's part of why we do the necropsy, to determine potentially what caused the death there. So there is a variety of reasons.
So then if there's a variety of reasons, did you have like an array of data where like some had really thin fat layers and some were healthier? Like that one that suffocated might have been more of a healthy sea turtle, theoretically?
So the one that did suffocate, she was extremely healthy, which makes it even sadder, because her fat was super thick and she actually had eggs still inside of her. So she wasn't done, laying her eggs. So that one was really sad. And then there's a lot of boat strike injuries. Some of them are fresher, so they are, thicker when we get them. And then others, you can tell that they've probably been hit a while ago because they were skinnier, so they were starting to wear away. So the cause of death sometimes does relate to their fat depth as well.
Great. Next question comes from Manatee is the name. Great presentation. Do you have any advice for anyone who's planning to major in marine biology? So I guess each of you guys can take a turn. We'll start with Skyler.
I think my biggest piece of advice is to get as much experience as possible. If you live in a coastal environment and can get hands-on experience doing what you're interested in, that's huge. For both Marisa and I, we got our start in sea turtles with through an internship with Dr. Annie Page Cargin, and that really just propelled me to keep on this path. So I think internships are very important for anyone interested in this field, and just getting to know people and getting as much experience as possible.
Very good advice. Marisa, do you have anything to add to that? I second that. Definitely get a lot of experience. You have to know that you love what you're doing before you immerse yourself in this field. And it is very rewarding in the end, but it is hard work. So another thing I would say is maybe like read up on like news and some literature, see what's out there already and something that you can contribute potentially, and see if this is a good fit for you. Great. Another question for the both of you guys. What is your favorite sea turtle species?
I'm pretty biased. I love the leatherbacks. They're the biggest. They're the most impressive. They dive the deepest. They travel the furthest. They're just very cool. They're literally living dinosaurs.
I like the green and loggerhead, surprise. I really like the green because they're kind of just like a hassle to work with, and I like that about them. They are strong and resilient, so I admire that. Oh my gosh, that's not what I expected for your rationale, but that's great. Let's see, we've got another question. This one is from Linda. She says, excellent presentation. I was surprised to see that fishing lines cause more issues than boat strikes. Wondering if there are current efforts to fish differently.
There are some, specifically for the leatherbacks, it's kind of hard. So a few years back, they created something called a TED, which is a turtle excluder device. So that's mainly for the smaller species. It's for trawls. It's like a little trap door that if a turtle gets in there, it can push through and escape, and so it won't drown. But with that, right now, they're not big enough to let leatherbacks through. So they know that they should fix that, but again, we don't really have it very well regulated, as well as they look at different hook types. There's certain hook types that they're starting to implement that may make it less dangerous to sea turtles, but again, we need more of these studies to really propel the management to find these solutions and make the efforts to change their habits.
Yep, definitely. Encouraging change is important. Next question here. So again, I think it's for Skyler. So for your research, you were using ATVs. Did those disturb the turtles at all that were nesting?
No, they don't peer very well, and the ATVs have red lights on them, which they can't really see. Since red is the first wavelength that's absorbed into the ocean over the millions of years they've been alive, they've started to not be able to see the red wavelength as well because it's not necessary for their life in the ocean. So whenever we're by the turtles or we're on the beach, we use red lights so it doesn't disrupt them. So no, we keep our distance with the ATVs too. As soon as we stay close to the water, so as soon as we see a track, we stop and then we'll walk up to the turtle, so we're not bringing this large vehicle next to it.
Got it. Another question. I walk along the beach and observe turtle shell fragments. Is it more likely that these turtles hatched successfully and made it to the ocean, or more likely that they were victims of predation?
It could be either one. it's hard to tell. There's a ton of eggshells that are left over when something hatches. So those honestly could be from the previous season. A lot of them end up getting just reburied in the sand. So it could be from any reason, honestly.
Next question here is, are there plans to use Marisa's research with the ultrasonography on live sea turtles?
Yes, that is actually something we're working on. I didn't include it in this part. I wanted to focus primarily on the ultrasonography of the carcasses, but we do, I think one of the images that I put in my presentation was me at the St. Lucie power plant ultrasounding one of the green turtles that was caught in their canal. So yeah, I do the live turtles, but I usually just do their dorsal shoulder region.
Okay. So we are wrapping up the questions. If anybody does have other questions, please feel free to ask them. I have a few more questions that I've thought of, but I can always ask them later on. So I want to get to your questions if you have them, but let's see, what did I write down? Oh, a question about how you draw blood on a sea turtle. Like, I know that when I get blood drawn, they're like looking for a vein. Do you do that with a sea turtle, or do you just sort of see what you get?
It's not as easy to see the vein, but you know the general location. So for where I take the blood from for the leatherbacks, it's very obvious. Since they're so big, the region where you can get blood from is also very big, so it's pretty obvious. We just kind of press around in their hind flipper region, and there's like a really soft patch where you know that's where the blood will come. For the other species, you do have to do it from the dorsal region, so you have to kind of poke around with your fingers a little bit. So we have a general idea of where to poke for each of those. It's not as obvious as when you get it done yourself and they have you like squeeze your hand so the vein pops out, but we still have a good idea of where to go.
Cool. Very good. Well, it looks like we've answered most of the public's questions for today. So thank you guys again so much for being here. Another round of applause for our wonderful graduate students. Thank you for kicking off our Marine Science Friday lecture series. And then for you all, please, we welcome you to come back again. We'll be back in 2 weeks on February 19th. With another talk about some of our charismatic megafauna. We're going to be doing sharks and rays, and the title of that presentation is From Taste Buds to Toxins, Understanding the Feeding Ecology and Toxin Transfer of Sharks and Rays in the Indian River Lagoon. So we hope to see you guys then. In the meantime, have a wonderful weekend.